Okay, guys, chapter 16, we're talking about the molecular basis of inheritance. So, we're going to be talking about, from a molecular level, some of the things that we have talked about already at a cellular level in this chapter. So, I want you to be thinking as you go through about making connections between this chapter and some of the things that we talked about in mitosis and meiosis. Alright, so before we get into talking too much about DNA replication and what happens to the genetic material, we want to talk a little bit about the history and how we got to where we are now and how we know what we know. So, it wasn't that long ago that we didn't even know that DNA was the genetic material. And so there was a big push, there was a lot of research being done, there were a lot of groups looking into trying to figure out what was the genetic material. And so basically there were two candidates for what could be the genetic material within a cell. Some people thought it was DNA, and some people thought it was protein. And so there was a big debate on that, and people were on one side or the other. There was a researcher at the time whose name was T.H. Morgan, and he showed that genes were located on chromosomes. And so because of this, the two components of chromosomes, both DNA and protein, became candidates for this genetic material. And so people started looking into this and trying to figure out, is it the DNA or is it the protein that's passed down from generation to generation? So in 1928, there was a researcher by the name of Frederick Griffith, and he actually discovered that it was the DNA that was the genetic material. And he did this by working with mice and two strains of bacteria. The two strains of bacteria that he worked with, one was pathogenic and one was non-pathogenic or harmless. And by pathogenic, we mean lethal or deadly. So the pathogenic strain would have been deadly. The other strain was harmless. So we're going to walk you through the experiment that Frederick Griffith did to demonstrate that DNA is the genetic material. Alright, so we said that he used mice, and he used these two strains of bacteria. So the first strain that he used were called S cells. So the S cells were the pathogenic bacteria. Or the deadly bacteria. So when he took live S cells and he injected them into the mice, the result was the mouse died. The other strain that he worked with was called R cells. So the R cells, excuse me, were the cells that were harmless. So they did not hurt the mice at all. And so when we injected the R cells into the mouse lived. There was no problem. So the next thing that he did was he took these S cells, which would typically kill the mouse, and he exposed them to heat. So he did what we call heat killing the cells. So that basically destroys the cells and made the cells so that when he injected them into the mice survived. So when he did the heat-killed S cells, the strain was no longer pathogenic. It was no longer deadly or lethal to the mice. So then what he did in experiment number 4, this was the crucial one, where he took a mixture of heat-killed S cells and living R cells and injected those cells into mice. So remember, this is a sort of a combination of experiments 2 and 3. And in both of these cases, in both of experiments 2 and 3, the mice lived. And so we might expect, or we might hypothesize, that if we just combine those two things, the mouse would live. But in fact, when he injected the mice with heat-killed S cells and living R cells, the mouse died. Not only did the mouse die, but he was then able to extract living S cells from that dead mouse. And so what this told him was that there was something in those S cells that transformed the R cells into lethal cells that killed those mouse. So there was some sort of transformative agent in those S cells that converted the R cells into S cells and caused them to be deadly. Now, there was a lot of debate about this experiment. A lot of people didn't believe it. A lot of people thought that Griffith was kind of off his rocker and didn't know what he was talking about. But so that was in 1928. In 1944, another group of researchers called Avery, McCarty, and McLeod came along, and they repeated Griffith's experiments and did some additional experiments. And they announced, based on their research, that the transforming agent or the transforming substance that Griffith talked about was in fact DNA. So they based this conclusion on the fact that the DNA worked, that only DNA worked in transforming harmless bacteria into pathogenic bacteria. So they looked at all the parts and pieces of those S cells to say what was it in those S cells that was able to convert the R cells into the S cells. And Avery, McCarty, McLeod said it was only the DNA that was able to do that. And so that told us that it is the DNA that was the genetic material. Despite this reaction, or despite this set of experiments, a lot of biologists remained skeptical. They still didn't believe that DNA was the genetic material, mainly because we didn't really know that much about it. Alright, but now we do know a lot about DNA, and so we're going to walk through the structure. A lot of this is probably going to be a review for you, but we will go over it in class as well. So we know that DNA has three main components. It has a phosphate group, it has a deoxyribose sugar, and it has those nitrogen bases that we call A, T, C, and G. So cytosine and guanine, thymine, and adenine. So if we were to look at the nucleotides here, so each of these structures that you can see here is a nucleotide. And so we have, I'll highlight them in different colors. Our phosphate group is red here. Our deoxyribose sugar, I'm going to circle in blue. And then our nitrogen base, I'll circle in green. So this one happens to be an adenine. So this is showing you the adenine base pair. But all nucleotides have these three features. And remember, on the sugar, it is this second carbon that no longer has the oxygen, that just has the H group instead of the OH group, and that's what makes it deoxyribose. We also know something about the base pairing of DNA. So we know that A always pairs with T, and C always pairs with G. We also know that DNA forms the double helix. So you guys have probably heard that before or seen that structure. DNA is always double-stranded, and it forms a double helix. And we'll take a look at that structure in just a second. But it's important to remember this base pairing and to recognize that A always pairs with T and C always pairs with G. That's going to be really important for us. Alright, so here's an image of that DNA double helix, kind of showing you what the double strands of that DNA look like. There's a very interesting story behind the discovery of the structure of DNA. So if you don't know this story, I highly recommend that you read up on about it. There's a really good PBS documentary called Photo 51 that I think you can find on YouTube that is really fascinating. So we all know that it is these two men, Watson and Crick, that received the credit for determining the structure of DNA. There's a whole backstory to that involving this woman, Rosalind Franklin, and how Watson and Crick really used her data in order to determine their model and based their model off of her data and sort of stole her data. So it's a really interesting story. If you're interested, I highly recommend this PBS special called Photo 51. But we're going to talk a little bit more about the structure and the parameters of this DNA double helix. Alright, so every time DNA replicates, we have to, every time a cell divides, we have to replicate the DNA. So DNA replication is a process that we've talked a little bit about in terms of mitosis and meiosis, and we know that it happens during interphase or during S phase. So that is when the chromosomes go from an unduplicated version like this to a duplicated version like this. So remember, we talked about both of these represent one chromosome. This is an unduplicated chromosome, whereas this one is a duplicated chromosome. So when we're talking about that chromosome duplication, we are really talking about DNA replication, and that's what we're going to talk about in the rest of this chapter. So again, this happens during S phase. It is necessary for cell division, and every daughter cell that is produced gets a copy of every gene. So this was our original DNA molecule. So all DNA is double-stranded, but this is our DNA double molecule, or our DNA molecule here. And when we make a copy of that molecule, we end up with two double-stranded molecules of DNA. And so this is what happens every time we go through S phase and prepare for cell division. And we're going to talk more about this process as we go through this chapter. Okay, so if we look at this a little bit, there are three main enzymes that are going to be important to us when we consider DNA replication. So here we have our original sort of parent strand of DNA that we are getting ready to replicate. The first thing that has to happen is an enzyme called helicase is going to come in and separate the two strands or unwind the DNA. So we get strand separation occurring between our two strands of our DNA molecule. The second enzyme that comes into play here is DNA polymerase. And the job of DNA polymerase is to attach complementary nucleotides. So what that means is that if I have a G in this position, DNA polymerase is going to put a C on my new strand. If I have a T in the second position, DNA polymerase is going to put an A on my new strand. If I have an A in this third position, DNA polymerase is going to put a T in my growing strand, and so forth. So DNA polymerase always attaches complementary base pairs, knowing that A always pairs with T and C always pairs with G. The last thing that happens is that the third enzyme, called DNA ligase, comes in and seals the two strands back together. So we reform those hydrogen bonds that are between these base pairs, and we now end up with two DNA molecules that look exactly like our parent strand. So that is the very general process of DNA replication, and now we're going to take a closer look. Okay, so the amazing thing about this replication process is that your cells do it with impeccable speed and incredible accuracy. It does take many more enzymes than the ones that we're going to talk about. We're only going to talk about those main three enzymes for now, but there are many more that are involved in the process, which you will learn more about later as you go through and take more upper division classes. But it is a complex process, yet your cells do it with amazing speed and incredible accuracy. Okay, so when we first talk about those two strands separating, so we have our two strands of DNA that look sort of like this. And the pen's not drawing really well. But the first thing that happens is we get that strand separation. So we might get something that looks like this. Okay, and so we end up with this area right here where the strands are separated. So we've got a wide gap here where we see strand separation. We call that the replication bubble or the replication fork. So that is where replication is going to originate. It's called an origin of replication, and that's where the two strands separate, and we end up with these replication bubbles. A eukaryotic chromosome can have hundreds or even thousands of these replication bubbles at any given time point. And replication is going to proceed in both directions along each strand of the molecule until the entire molecule is copied. So that creates a layer of complexity for us that we have to think about and work out, which we're going to talk about now. All right, so let's take a look a little bit more closely at these replication bubbles, or the replication forks that we see. So here's our parental strand of double-stranded DNA molecule, and we're getting ready to carry out replication of this molecule. So this cell is getting ready to undergo division, and so we are replicating or duplicating all of the chromosomes, or all the DNA inside the cell. So we get these replication forks where the two DNA strands have separated. So sometimes they're called bubbles, sometimes they're called forks. It pretty much means the same thing. And so we get strand separation, and then we get synthesis of these strands occurring in both directions. And so that's going to be important for us as we go through this process, and to know that the DNA molecule is being synthesized in in opposite directions on each of those strands at the same time. And so we end up with then our two daughter DNA molecules being produced. Notice that when we show the colors here, our old strand, we have two kind of dark blue strands representing that DNA molecule. And when we look at our two newly made DNA molecules, one strand is dark blue and one strand is light blue for each of these new molecules. That represents that one of those strands is sort of our old strand, or our parental strand, and the other strand is the newly synthesized strand from the DNA polymerase. So each DNA molecule contains one old strand and one new strand, and that's going to be important to remember as we move forward. Alright, so DNA polymerase, remember, that's the enzyme that brings in the complementary nucleotides, and that is going to add bases to the DNA strand at the replication fork. So remember, if we have this sequence in our parental strand, so that's our kind of our old strand, or our parental strand, we would get the DNA polymerase would bring in T, A, G, and C to sort of base pair with those. So it's bringing in the complementary nucleotides. And it does this at a rate in bacteria of about 500 nucleotides per second. And in human cells, it does it at a rate of about 50 nucleotides per second. And that is pretty fast, pretty incredible speed considering the complexity of the DNA molecules inside these cells. Okay, so DNA has what we call an anti-parallel structure between the two strands of the helix. That means they run in opposite directions. And right now, I don't want you to worry too much about what 5' and 3' actually means. I just want you to know that one end of the strand is designated 5', and the other end of the strand is designated 3'. So this strand, for example, would be running in the 5' to 3' direction. On the other strand, it's running opposite. So up here we have the 3' end, and down here we have the 5' end. So this guy, if we're talking 5' to 3', is running in the opposite direction. So that's what we mean by anti-parallel DNA strands. They're running in opposite directions relative to that 5', 3' end. And so we do determine that based on the structure of the sugar. But again, I don't want you to worry too much about that. I just want you to understand that they are running in opposite directions, and therefore it's what we call anti-parallel. Okay, so this anti-parallel structure actually affects the way that DNA is replicated. And the key here, if you don't remember anything else from this chapter, you have to remember this point. The DNA polymerase can only add nucleotides to a free 3' end. So that is so important. So this is a little bit hard to see. For some reason my program got edited here. But that should say 3'. So DNA polymerases add nucleotides only to a free 3' end of a growing strand. And therefore a DNA strand can only be elongated in the 5' to 3' direction. So that's the only way that we can grow a strand, is in the 5' to 3' direction. We can only add new nucleotides to these 3' ends, and that is so important to remember as we talk about the two different strands and the process of replication. Alright, so we're going to look at antiparallel elongation and how this functions on each of these strands. So we have our two parallel strands here. Our strands are running 5 prime. This strand is running 5 prime to 3 prime in the right direction. This other strand is running 5 prime to 3 prime headed to the left. So you can see how they're oriented in opposite directions, or what we call anti-parallel. Okay, so the first thing that is going to happen when we're getting ready to carry out DNA replication is we're going to get separation of our strands. So DNA helicase, remember, is the enzyme that is going to come in and separate those two strands and create the replication bubble. So DNA helicase has come in and unwound or separated our two DNA strands. Okay, so we're going to talk about this strand on the bottom here first. This is called our leading strand. So remember, this is the one running 5 prime to 3 prime, sort of in the left-hand direction here. So when we make our new strand, our newly synthesized strand, again, it has to be anti-parallel. So our 5 prime end would be down here on the left-hand side, and our 3 prime end would be up here on the right-hand side. So this would be the 3 prime end. If we're just talking about this segment right here, this is our 3 prime end. And remember, we can add nucleotides to a free 3 prime end. So here's our DNA polymerase. It's going to move down our strand heading to the right. So the green arrow here represents the direction of movement of DNA polymerase. And so as it moves down that strand, it can add complementary base pairs as it goes until it gets all the way to the end and has copied its entire section of the DNA molecule. So we are just consistently moving down this molecule, adding nucleotides as we go to the 3 prime end. So that, remember, is referred to as the leading strand. The lagging strand is a bit more complicated, and I'm going to show you a video of this in class that doesn't post well here in this format and this medium. So I will go over this with a video in class, which I think will help you very much. But we require the use of RNA primers when we're talking about the leading strand or the lagging strand. And that helps us know where that DNA polymerase should bind. So the idea here is, remember, we can only add nucleotides to a 3 prime end. So DNA polymerase is going to move down the strand in to the right here, so direction to the right. And as it does that, it can add nucleotides as it goes. The problem is it's going to run into another primer right here. And so that DNA molecule is going to have to jump backwards. And again, this is a little bit tricky. And so I'll show you this in class, and I'll walk you through this better in class. But there are jumps that DNA polymerase has to make in the lagging strand, which make it a bit more complex of a situation. And so what we end up with then are fragments of the DNA, the newly synthesized strand, and these fragments are called Okazaki fragments. And so it is then the job of DNA ligase to come in and seal these fragments. So what happens is the RNA primers are going to get replaced with DNA, and then DNA ligase is going to come in and seal this newly synthesized strand so that it is one complete strand. So we will go over the processes of leading strand and lagging strand synthesis more in class when I show you the video, and I think that will help it make sense to you. Alright, there are also many good videos on YouTube. So if you just search for DNA replication videos, this is one example of one that you could watch, but there are many, many more on YouTube. So good luck with chapter 16, and I will see you in class.